So I just told you about Ki, but you can imagine that's going to depend on the K off. We're going to have those little K's, the rates of off and on, and I told you before that those can be pretty hard to get. So another way that we think about this is in terms of IC50s. So IC50s are the same as EC50s. Basically, but it's the concentration of inhibitor that produces 50% enzyme inhibition. And so you measure it in the presence of a constant substrate concentration. So when an enzyme binds to the active site, it's going to be a competitive inhibitor. So that means that it blocks the binding of the substrate. It can also bind to an allosteric site, and we have that non-competitive inhibitor. So just as you would imagine, right, because like I was showing you before, when you're having complex equilibria here, and we're not actually getting rid of the substrate, that's not how enzyme inhibitors work. And that means that because of this, we need to keep this high and this low, right? The amount of enzyme inhibitor complex high, the amount of enzyme and substrate complex low. And so what ends up having to happen, because that drug's getting metabolized, etc., is that we have to administer drugs daily to multiple times a day to maintain the enzyme in that complex. So we learned, this might sound really familiar to what we talked about before with receptors, and that's because it is analogous. Receptors, we talked about agonists and partial agonists and antagonists. And here for enzymes, this would be a good substrate versus a poor substrate versus competitive inhibitor. So these are not, you could make this analogy a different way, I will tell you that much, but this is an internally consistent way of making this analogy, so that's always good. So there's really 5 approaches to designing competitive reversible inhibitors. So this is helpful, right? There's simple competitive inhibition, stabilization of an active enzyme conformation, that's number 2, alternative substrate inhibition, transition state analog inhibition, that's going to be important when we talk about antivirals, and slow tight binding inhibition. So this is the simple competitive reversible inhibition is the most common type. So the inhibitor rapidly binds and reversibly binds to the active site. And frequently what you do is that it is the target looks like either the substrate or the product of the reaction. So the target looks like the substrate, or the inhibitor looks like the substrate or the product of the reaction of the target enzyme. So these can be, some of these are growth factor. So these are anti-tumor drugs, and they inhibit kinases. Again, we can see that because they have the IB there, but they're anti-tumor agents, and that what they're doing here is that they're inhibiting one of the activities of a growth factor receptor. So they're not blocking receptor binding. In this case, they're blocking the signal that the receptor gives to the rest of the cell to tell it to grow. So you could imagine that's really important for cancer drugs. So we've actually gone through this earlier in the slides when we were talking about pharmacophores and drug development. But kinases are really important to cancer treatment. And kinases lead to uncontrolled, can also lead to uncontrolled cell proliferation if they're not properly controlled. So kinases occupy a lot of key checkpoints in the cell cycle as the cell undergoes its growth. And so if some of those key kinases are mutated so they can't, there's not the same checks and balances that would normally exist when a cell grows, the cell can grow unchecked. And as we've talked about before, that epidermal growth factor, it's a growth factor, and so it's important in a lot of cancers because if you can mutate that, and in this case, what you end up doing is you hit these kinase domains that are sending the signal inside the cell. And so what the kinases do is they're going to phosphorylate another protein, and this is going to amplify until that signal gets from the outside of the cell to grow, gets to the nucleus, and then genes are transcribed in the nucleus that make proteins that get the cell to grow. And so what we're doing is we're blocking this signal going to the nucleus to get cells to grow. So in this case, the way that they did this, an example of an anti-cancer drug that was done by Zeneca, now AstraZeneca, is they had a couple of hypotheses, and so they want to mimic the substrate of the kinase in the epidermal growth factor, which is a tyrosine. So they're going to mimic the phenolic ring. They're going to mimic the hydroxyl group in tyrosine. So they want something that's going to be like a hydroxyl, so it's an oxygen or a nitrogen. And they need to mimic a little bit of ATP. And so they found different drugs that work pretty well, and they all look similar. So in this case, this is from Zeneca, so that's what's from their screen. That's ended up being how it looked. So they have kind of their tyrosine group and their ATP type group. And all these groups tend to have something that looks kind of like their tyrosine and kind of like their ATP. And they all kind of bind the same way. So they bind both to the tyrosine and the ATP binding sites. So all these drug companies didn't have access to the same data, but they had the same kind of concept, and all their drugs ended up looking similar. So another example of this is a really important drug called Imatinib, and it's an anti-leukemia drug, so for chronic myelogenous leukemia, or CML. And so this is where you get it's excessive proliferation of bone marrow. Bone marrow is what makes your white blood cells, so that's why it's leukemia. So excess of white blood cells in this case. And what happens is that you get two parts of chromosomes, two different pieces of chromosomes. There's a specific way that they can kind of stick together, and during non-homologous recombination events can stick together and form a mutated chromosome. And this chromosome leads to the expression of a protein called BCR-ABL. And this... So basically, it's a fused chromosome. Two pieces of the chromosome are fused, and it changes the kinase inhibitor. That changes that kinase, that ABL kinase. It changes its activity, and this causes CML. So if you have CML, if someone has CML, then what they can do is karyotype the white blood cells, so they can look at the chromosomes of the white blood cells, and if their chromosomes have the specific Philadelphia chromosome, they can say, okay, this person has CML, and they have CML caused by this specific mutation that leads to this protein BCR-ABL, and we know how to inhibit this protein, so this particular type of CML can be cured. So this is really exciting because it's like, okay, this is a mutant kinase, so it's not a kinase that we normally have. So that's a great drug target. So Novartis took this on, and so they ended up developing Imatinib. And notice what they had to do, right? So they've started with this as their lead, but it wasn't soluble. It wasn't soluble enough to cross the gut. It was too hydrophobic, and that's bad too, because not only does it not cross the gut, but in the liver, stuff that's really highly hydrophobic will get metabolized right away in the liver. So what they did was they attached this group. So it's not aromatic, it's got a couple of nitrogens, it's pretty basic, and this helped it to be a lot more water soluble. And so this is the final Imatinib from their lead. So they've also crystallized it, and they've shown where things bind. And in what's happening is that this mutant kinase. This is the conformation of the mutant kinase. And what happens is that the mutant kinase binds Imatinib, and when this happens, it's stabilized so that it can't bind ATP. So the kinase here, there's ATP bound. It needs to bind ATP in order to do the rest of its reaction. Imatinib stabilizes it in a conformation, binds in the active site, and stabilizes the active site in a conformation so that it can't bind ATP anymore. So it's stabilizing an inactive conformation. So instead of competing with ATP, you see it's stabilizing. So you see this active site this part looks pretty much the same. These green loops here, they look pretty much the same. But notice where this helix is, and this loop here, this loop that's non-structured here, it's in red, here it's in blue, it's all flipped over, and basically this protein isn't able to bind ATP where it would and do catalysis. It both isn't able to bind ATP and also isn't able to do catalysis. So this is right, the BCR fuses to ABL, and when it's this fusion protein, the ABL is always active, and so the cell's always proliferating. Okay, transition state analogs and multisubstrate analogs. In this case, the enzyme binds to the substrate tightest at the transit. Well, this is taking advantage of the fact that the enzyme binds to the substrate tightest at the transition state. So here's an anti-tumor natural product drug. So you can see how this looks like adenosine, and it inhibits adenosine deaminase. So this is your body's way of processing adenosine, as in the base that's in ATP. And so you can see that you would likely need this kind of catabolism for cells to live. And what happens is this inhibits it incredibly well, and that means that it's incredibly toxic. So this becomes an incredible toxin, which is really good for use as a chemotherapeutic. So what they think it's doing is that it mimics this transition state. So this is the starting material is ATP basically, or it's not ATP, it's just adenosine without the phosphates on it. It's not even adenosine monophosphate anymore. You're breaking down adenosine. This used to be ATP over here. And you're breaking it down. And what you're doing is you break it down, just like when we catabolize drugs, is that you're starting to add more hydroxyls and oxygens on there. In this case, you're pulling off this amino group and you're putting in the hydroxyl group. So what the drug does is it looks like the transition state. It mimics this, and remember, enzymes bind most tightly to the transition state. So it mimics that because it's got this hydroxyl pulled over there in the same way. So all this transition state is all chelated to that zinc 2 plus. So in this case, so in lymphocytic leukemia, so a different type of leukemia, so that's lymphocytes. Before we were talking about myelogenous cells, so it's a myeloid cell. These are T cells that are made in your thymus, so it's a different type of leukemia. This activity, this enzyme activity tends to be elevated, and so you're inhibiting this, and hopefully hitting those leukemia cells over your regular cells, especially because this is so toxic. Okay, slow tight binding inhibitors. So the enzyme inhibitor equilibrium is reached slowly. Basically, they are tightly binding, but the complex formation can take a long time. So some of the examples of this are going to be your ACE inhibitors, and so these are like lisinopril. These are common antihypertensive drugs. And what they're doing is that they're telling essentially the smooth muscle that lines your vascular endothelium to not contract as much. So your veins are not as rigid, and so the pressure's lower. So what happens is that we're going to try to inhibit of the vasoconstrictor that produces the enzyme that produces this vasoconstrictive hormone. And so what we're going to do is inhibit the production of this hormone that tells your veins and arteries to constrict. And so the way that this worked was that this is working by catalyzing the hydrolysis of vasodilator bradykinin, just so you know. So they originally found this in pit viper venom back at what is now Bristol Myers squid. And so they looked and found that it was a zinc-containing enzyme. They figured out the mechanism of action. That they had this hypothetical active site that they came up with based on this peptide hydrolysis mechanism. And what they did was they made models of the substrate. So they came up with this. They're like, okay, it's getting hydrolyzed. The enzyme's got a zinc 2 plus. Maybe the substrate looks something like this. So with this, they used a peptidomimetic, like we talked about before. And with that inhibitor, the structure they had before. And they used that peptidomimetic because it was orally unstable. Otherwise, it's a dipeptide. But there were only weak inhibitors when they tried that trick. So what they did was that they got rid of this carboxylic acid and made it, that carboxylic acid and made it something that was going to coordinate to zinc better. So they had this guy, coordinates zinc pretty well. They changed it to this guy, awesome zinc coordinator. And now they're able to finally start to get good. This is what they came up with finally. So they have a really good inhibitor, the Ki, this is captopril. And they were able to do some structural modifications. So this is basically, you can see it's kind of going the reverse way till they got to captopril. So basically, they couldn't get anything better than captopril. Every change they made messed with the Ki. And so this is like those tables we've made before. Here your conclusion would be every part of captopril is important for binding. So this is the first ACE inhibitor. The first ACE inhibitor. And now we're going to talk another building on this ACE inhibitors is talking about combination therapies. So what they do now is that you work with the water retention caused by aldosterone, and also you inhibit that bradykinin breakdown. So you have less water in your veins or less water in your arteries. You have less water in your circulatory system, so that's going to lower the pressure. You are inhibiting the bradykinin breakdown, and so the combination of the two is going to lower your blood pressure, and you can also use a beta blocker as well. So there's two side effects. Rashes and loss of taste. Yeah, I mean, they could probably market it as a diet drug, but, you know, they didn't ask me. So they thought this might be due to the thiol group. You know, thiols have some problems. And so when they had to go back off of the thiol, and they increased the potency. So you're saying, okay, well, here's the question. From our structure here, we know that every piece on captopril is important. So if we turn this back into COOH, right, it's going to turn it back to here, and that sucks. So what are we going to do? What they did was they added more spinach to make this interact with the enzyme more. They added more structure. So they added, to increase the potency, they added more interacting sites to interact with that enzyme. So remember, captopril is really small, so they can add more stuff on. So they started adding groups to counteract the hydrophilicity. But they found in this case what they had, their original lead was that they had poor oral absorption, and they fixed that by adding an ester. And they ultimately came up with a group like this. This is lisinopril. So you can see how this is related to captopril, right? Now they have this other group here to more interact with the enzyme. They've got that carboxylic acid there to bind with the zinc. Another example is levastatin. So we've talked about the levastatin when we talked about HMG-CoA and inhibiting the formation of cholesterol to form atherosclerotic plaques, and that also hardens and narrows the arteries. And here, it's the rate determining step in cholesterol biosynthesis, which is a huge pathway. But what they're able to do is take something that looks like this transition state here and make an inhibitor. So it's connected, HMG-CoA is connected to this coenzyme A, which you just talked about from pantothenic acid. And we have HMG, that HMG group. And so what we're going to do is find some transition state inhibitor. And there is a natural product that they found from a fungus in this case. And so this is what's called levastatin. And it has groups similar to, that look similar to that transition state. So this transition state here. So it's a really potent inhibitor, so the Ki is really small. So that's a good thing. And it mimics this intermediate by this structure. So what they did is that they wanted to make more statins, and they can start playing around with some of the other groups, right? They check and see what's in the pharmacophore. In this case, when they got rid of this methyl group, they got simvastatin. So that's even more potent. There's also type 2 statins, and we talked about this before because one of those is a a parole-centered drug. Okay, so now we're going to talk about irreversible inhibition. So irreversible inhibition doesn't mean you permanently lose the enzyme in your life. You don't lose the gene, you lose the protein. And so the thing is, protein biosynthesis can take a long time. So there are irreversible inhibitors for a lot of different drugs. They're just not as commonly prescribed, except for proton pumps for acid reflux. And so that means that they're used because they're used for acid reflux. And now a lot of these are no longer, these are off label now. These are, you can buy them over the counter. Except for this is a platelet inhibitor, blood thinner. So of these irreversible inhibitors, there's two general types, and that's infinity labeling and mechanism-based enzyme inactivators. And one of these that's important is an anti-tumor agent known as 5-fluorouracil. So basically what this does is that it's going to inhibit DNA biosynthesis. So you can imagine that cancer cells, because they're growing more quickly, every cell that's the daughter cell of these cancer cells is going to need DNA, right, to live. And so if you can inhibit its ability to produce DNA, the cell is going to end up dying, and it's not going to be producing any daughter cells. And so hopefully you're definitely going to hit every quickly growing cell in your body, as you do with most of these chemotherapeutics, which is why you lose things like your hair. Have a lot of other side effects, but it will hit the cancer cells more preferentially than other cells in your body. Okay, so what you can do is you have these basically fluorinated drugs that look like DNA bases. And so what happens is that your body metabolizes it so 5-fluorouracil is not active. Your body turns it into the active form of the drug, and it gets incorporated into your body. And when it does, it messes with nucleotide synthesis. So what it does is that it makes it so that your body thinks this is a thymidine. So GCAT, it thinks it's the T of your DNA base. So it goes here, your body turns it into this. It starts off here. Your body is like, oh yeah, this looks like some good stuff, you know. And your body, it can also go through these pathways here. Your body then takes this and says, this is a T. And it incorporates this. It tries to incorporate this into the DNA that the thymidine synthase does, but it inhibits that ability to do that. Okay, so selective toxicity, this is something we've been talking about, is that most tumor cells replicate more quickly than regular cells. And 5-fluorouracil is taken up into cells more efficiently than into most normal cells. So you have a couple of pieces that are helping you here. But it really does destroy rapidly proliferating cells, and that includes So that it really does include a lot of cells in your body, so it can be very challenging to take, even when it's taken externally, say for skin cancer. So when thymidine synthase, again, and notice that thymidine synthase is using this dihydrofolate reductase. And it's doing, oh my god, the vanilla gets Michael. Oh, so cool. And so what happens here is that you have, you're forming a covalent intermediate between the cofactor, tetrahydrofolate, and the fluorouracil and the enzyme. So you got the enzyme, you've got the cofactor, and you've got the fluorouracil all forming this cross-linked protein that's not doing any catalysis. It's not going to do anything. Dang it. And with that, we'll end this lecture.